Hello dear friend and learners, myself Mr. Mohammad Sadiq, lecturer of Kamla Nehru Polytechnic Pharmacy, Aurangabad. Today we are learning the process of metabolism of carbohydrate, that is the glycolysis. Learning objective of this glycolysis process: first, to understand the glycolysis process, then what are the enzymes involved in the glycolysis process, and in which step the energetic, that is the ATP, will be involved and utilized and formed. First, there is one question: why there is a need to understand the glycolysis process? Before operating to this video, you have to just stop and write your answer on your notebook. Okay, now when we are concluding that the glycolysis is the one only process when we without oxygen we can convert glucose into the pyruvate units. That's why we have to understand the glycolysis process. And for diploma student, it is the in that syllabus. That's why they have to study the glycolysis process. What is glycolysis? Glycolysis is a catabolic process. That is, glucose will be break down into pyruvate molecule by the oxidation process, where there is so many energy formed in absence of oxygen. That's why the process is called anaerobic fermentation of glucose. The first molecule which is utilized in this process is a glucose, which contains 6 carbon atoms, and then in that process the first step is hexokinase catalysis, which is known as the phosphorylation process. That is addition of phosphate into the glucose molecule at the 6th position. That is, glucose will convert it into glucose 6-phosphate in the presence of hexokinase enzyme by the utilization of 1 mole of ATP to ADP. For conversion of ATP to ADP and the addition of glucose to glucose 6-phosphate, it requires a hexokinase enzyme and the magnesium ion process. Magnesium ion. It is the spontaneous breakdown of addition of glucose into the glucose molecule, that is the addition of phosphate molecule, which is called the induced mechanism, in which the enzyme is induces by the glucose molecule, that is the substrate, and the ATP. And the second step, which is to be known as the isomerization, that is glucose 6-phosphate will open the chain and convert it into fructose molecule, that is the ketone molecule, and will form the fructose 6-phosphate in the presence of phosphoglucose isomerase. The third step, which is the next one, is the phosphorylation. Another is the phosphorylation. In that third step also, the ATP molecule has been utilized in the presence of hexokinase enzyme and the magnesium ion, and this ATP will converted into ADP. As in the first step, glucose 6-phosphate will be formed, and the third step, fructose 6-phosphate is formed, is converted into fructose 6 diphosphate molecule. That is, in this step, at the first position of fructose molecule, phosphate will be attached from the ATP molecule. And the next step, that is the breakdown of this fructose 1,6 diphosphate molecule into dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. As fructose contains 6 atom, 6 molecule, 6 carbon atom, they will be split into or cleave into 3 carbon atom as dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. The most important is the glyceraldehyde 3-phosphate, which is forwarded into the next glycolysis process, in which also and when we are seeing this process, the fructose 1,6 diphosphate will be cleaved by the enzyme of aldolase, and this dihydroxyacetone phosphate will also be converted into glyceraldehyde 3-phosphate with the enzyme of triose phosphate isomerase. And as we know that the hydroxyacetone phosphate just not on only one molecule, it will be converted into 2 mole, that is dihydroxyglyceraldehyde 3-phosphate, also converted into the dihydroxyacetone phosphate. So next step, when we are seeing glyceraldehyde 3-phosphate will be converted into the 1,3-diphosphoglycerate, which is the oxidation process, as we call this is the oxidation and addition of oxygen. Here in the 5th step, the energy will be formed, that is energy will be released, that is NAD will be converted into NADH, and this will be converted, that is glyceraldehyde 3-phosphate converted into 1,3-diphosphoglycerate with the help of glyceraldehyde 3-phosphate dehydrogenase enzyme, and that 1 mole of NADH will be formed, that is 3 ATP will be generated. Next, the transformation, that is 1,3-diphosphoglycerate will be converted into 3-phosphoglycerate. In here also the ATP will be formed in presence of ADP, and with the help of phosphoglycerate kinase, 1,3-diphosphoglycerate kinase will be converted into 3-phosphoglycerate kinase in the presence of phosphoglycerate kinase and the magnesium ion. Here the 1 ATP molecule will be generated. Next one, the phosphoglycerate mutase, that is 3-phosphoglycerate will converted into 2-phosphoglycerate, that is they will just changing the position from the hydroxyl onto the C3 to the hydroxyl onto the C carbon atom. When we are seeing the next step, that is the anolyze catalysis process, 2-phosphoglycerate will be converted into phosphoenol pyruvate, that is the removal of hydrogen, and then the second in the they will form the enolate intermediate, and this enolate intermediate will release the hydroxyl group from the carbon atom, and they will form the phosphoenol pyruvate, and this dehydration reaction is magnesium dependent, in that 2 magnesium ion has been utilized, and they will form the 1 mole of water molecule. Next one is the pyruvate kinase catalyze reaction, that is phosphoenol pyruvate will convert into the pyruvate molecule, that is they will just release the 1 phosphate molecule from the carbon number 2, and they will form the next ATP molecule, that is ADP will be converted into ATP with the help of pyruvate kinase. Here also the magnesium ion also required. When we are seeing in detail, the phosphoenol pyruvate first converted into enol pyruvate, that is we just changing the hydrogen position for by removing the phosphate molecule to the ATP to the ADP, and will form the ATP, and then add the hydrogen molecule to the enol pyruvate to form the pyruvate, and this require inorganic cation, that is potassium and magnesium bind to the anionic residues at the active of the pyruvate kinase. When we are looking into the overall process, that is glucose will convert into the glucose 6-phosphate by the enzyme of hexokinase, where the 1 mole of ATP has been utilized, which will convert into ADP. Then glucose 6-phosphate will convert into fructose 6-phosphate with the help of phosphoglucose isomerase. Then fructose 6-phosphate that will be converted into fructose by 1,6-diphosphate with the help of fructose phosphofructokinase in the by the removal of by the utilization of 1 mole of ATP. This 1 fructose 1,6-diphosphate will split or cleave into the glyceraldehyde 3-phosphate and the hydroxyacetone phosphate in the presence of aldolase enzyme. This dihydroxyacetone phosphate will also be converted into glyceraldehyde 3-phosphate, and then this fructose 1,6-diphosphate give the 2 moles of glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate will convert into glyceraldehyde 3-phosphate with the help of triose phosphate isomerase. This glyceraldehyde 3-phosphate then will give the 1,3-diphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase, and they will give the NADH molecule, that is 3 moles of ATP will be generated. This next 1,3-diphosphoglycerate will convert into 3-phosphoglycerate, that is that the one position phosphate molecule will be removed to the and added to the ADP, and they will form the ATP in the presence of phosphoglycerate kinase. These 3-phosphoglycerate kinase just changes the position, that is 3 from 3 to 2 phosphoglycerate with the help of phosphoglycerate mutase. These 2 phosphoglycerate will release the water molecule and will form the phosphoenol pyruvate, and with the help of enolase enzyme, this phosphoenol pyruvate will convert into the pyruvate molecule, and in this step also ATP will be generated with the help of pyruvate kinase. When we are looking to the next, this is the overall and glycolysis process, but we are seeing to anaerobic fermentation. This pyruvate in absence of oxygen will convert to lactate dehydrogenase, where the ATP and NADH will be utilized, and this lactate dehydrogenase will further move on. This pyruvate molecule will go on to the TCA cycle, and this started the breakdown to carbon dioxide and water molecule. Okay, this is all about the glycolysis. Thank you.